Our group will be discussing HIV, or human immunodeficiency virus. I'm Bethany Benson, and I'll be starting the presentation off discussing etiology. And HIV, it contains two identical single-stranded RNA or positive ssRNA. HIV is actually a retrovirus, meaning the virus has the ability to reverse the usual flow of genetic information, which is normally from DNA to RNA. Outside the cell, a retrovirus exists as a single-stranded RNA genome packed into a protein capsid, along with a virus-encoded reverse transcriptase enzyme. During the infection process, the viral RNA enters a cell and is converted to a double-stranded DNA molecule by the action of this crucial enzyme, which is able to polymerize DNA on either an RNA or a DNA template. HIV is a round ball-shaped virus and is surrounded by a cell envelope. In order to replicate, HIV attacks and destroys the CD4 cells of the immune system. And CD4 cells are a type of white blood cell that play a major role in protecting the body from infection. HIV uses the machinery of the CD4 cells to multiply or make copies of itself and spread throughout the body. This process, which is carried out in 7 steps or stages, is called the HIV life cycle. And here in this image, we can get a better look at the HIV life cycle. We can see here at the top, in the first step, the fusion of HIV to the host cell and all the way to the end of the cycle with new immature HIV being formed and spread to other cells. I'm not going to go through all of this, but if you'd like, you can pause the video here and take a picture of this image. And there are several simple features, several special features of HIV. The first is the ability to fuse host cells, which is what we just briefly went over in the HIV life cycle. But in order to enter the cell, HIV has to fuse with the host cell to insert its viral genome into the host cell. The second ability is the latent phase, which we'll talk about in the next section. And lastly, HIV has 3 stages of progression: acute HIV infection, chronic HIV infection, and AIDS, which also will be discussed later on in the presentation.
Awesome. Thank you, Bethany. My name is Hans, and I'll be presenting the reservoir portion. And to begin, disease reservoirs, or reservoirs of infection, are the population of a pathogen or the specific environment in which an infectious pathogen naturally lives and reproduces. In other words, the reservoir is a location in which pathogens primarily depend on for their survival. Latent HIV reservoirs can be found through the body, including in the brain, the lymph nodes, blood, digestive tract. A latent HIV reservoir is a group of immune cells in the body that are infected with HIV but are not actually producing new HIV. Bethany had just explained the production of HIV. However, like she had also mentioned, some HIV-infected cells go into a resting or latent state. And while in this resting state, the infected cells don't produce new HIV. When HIV infects cells in that way, it can hide inside these cells for years, forming a latent HIV reservoir. At any time, cells in the latent reservoir can become active again and start making new HIV. Now I'm gonna start touching into the signs and symptoms. And to begin with, it's important to understand that there's a difference between signs and symptoms. Signs are objective evidence of a disease as noted by an observer, such as a rash or a fever. Symptoms are subjective evidence of a disease as sensed by the patient, such as aches, pains, and fatigues. Common signs of HIV, and I'm just going to name a few. You can also see some on the little picture right there on the slide. But sore throat, getting bad infections a lot, headaches, losing lots of weight quickly, bruising more easily than normal, having deep or dry coughing spells, skin rashes, the list goes on. However, something to note about symptoms of HIV is that you cannot really know unless you actually go get tested. About 40 to 90% of people have flu-like symptoms within 2 to 4 weeks after HIV infection. Other people do not feel sick at all during this stage, which is also known as acute HIV infection. Again, Bethany had touched on that. Early infections is defined as HIV infections in the past 6 months recent and includes acute, very recent infections. Now, to the progression to AIDS. If you have HIV and you are not on ART, eventually the virus will weaken your body's immune system and you will progress to AIDS, the late stage of HIV infection. Symptoms to this can include rapid weight loss, recurring fevers or profuse night sweats, extreme and unexplained tiredness, sores of the mouth, anus, or genitals, memory loss, depression, and other neurological disorders, and the list also continues. But now I'm going to pass it over to my friend Samir, and he'll explain how the disease is transmitted.
Hey, good evening. Thanks for that, Hans. Appreciate that, and thank you for that, Bethany. So now we're just going to talk about how HIV is transmitted, how it's received. So the way HIV is transmitted is through multiple and various ways. One is which HIV is transmitted through the exchange of bodily fluids from a person who is a carrier of the virus or of HIV. Another is the example of mother passing to child during childbirth. Yet again, another example would be the sharing of medication or drugs by needle. HIV can also be transmitted through sexual acts and also through blood and organ transplants, which is less common more so now due to our medical advances and our adjustments and our sanitation of our medical equipment and of our procedural and our procedures that take place during those surgeries. Now, so what actually happens with HIV and how it's transmitted is that it usually finds its way into the mucous membrane. And so for those of you who don't know, mucous membrane can be found in the rectum, the vagina, the opening of the penis, and even the mouth. And so what happens is when the virus comes into contact with those body parts or any body part that has some mucous membrane, it is a little more permeable to get through and to navigate through the body and even to get into the bloodstream. Another way that that HIV is contracted is, as I mentioned, is through the bloodstream, right? Through the sharing of a unsanitized needle of someone who may have the virus, and then that person is injected with a drug or medication, and then that needle is not then sanitized or not properly disposed, then giving it to someone else who receives the injection after that. And so because HIV is so deadly and it is so powerful, as you can see at the bottom of my slide, there are some visual aids that really kind of water and really dull things down as to how you can and can't get HIV. So with that being said, I wanted to move to the prevention slide, which I was also responsible for. The key thing to note, and something that will be touched upon in later discussion, is that HIV there is no cure for HIV. So the following methods that I'm going to discuss are very important and critical that we make sure that our practice and that we make sure that we are well aware of, because, as mentioned again, there is no cure for HIV, unfortunately. So with that being said, some of the prevention methods that are effective from preventing HIV is practice with safe sex, such as testing if you and your partner have HIV, using condoms. And I love how we're at BYU Idaho and we can talk about the gospel and intertwine it with our lessons and curricular. And so from a gospel standard, live the law of chastity, limit the number of sexual partners that we have. And when taking drugs, if we have the option to, don't take it via needle or injection, but other methods. And if there's no other way to receive the medication, then make sure proper sanitary precautions are first taken, and then take pre-exposure prophylactics or PrEP drugs if living with a high-risk lifestyle, if you're currently sexually active with a person who does have HIV, to prevent those things.
Awesome. And then I wanted to talk about treatment. And treatment is really, a tricky portion just because there is no cure for HIV, just like Samir just said. So a website called Fresh Quotes said, "Prevention is better than a cure, especially when there is no cure." So the best way to deal with HIV is to just never get it because there's no way to cure it. And the only thing we can do is reduce the effect it has on the body. So the best way health professionals have found to do this is through antiretroviral therapy, which is commonly known as ART. ART is a combination of drugs that reduces the viral load of HIV in the blood, meaning that there's less of the virus in the blood. And it allows the immune system to partially recover and fight off some of the infections that it otherwise would not have been able to. ART also reduces the risk of transmitting HIV to other people, which makes it great as a preventative measure as well for others. And like any drug, antiretroviral therapy drugs do have some side effects, but those side effects have been concluded to be better than the consequences of not taking the drugs at all, which is having your HIV progress to AIDS and then going to death. But then we kind of wanted to talk about the history of HIV. It's so crazy and full of misconceptions, prejudices, and more. So we're just going to cover a little bit of that. So I primarily decided to focus on the disease in the US, but we'll start out in the early days. So the virus started out as simian immunodeficiency virus, or SIV, and was found in chimps and monkeys. It's estimated it was transmitted to humans as HIV in the 30s through bushmeat hunting, in which hunters would track and kill chimps and monkeys for food and come into contact with their blood. And they're pretty sure that the virus came to the US in about the 1960s. AIDS itself was actually discovered a year before its cause, HIV. So since AIDS is the actual cause of death, it was easier for medical researchers to identify it than it was to find HIV, which, as we showed, doesn't have a lot of signs or symptoms because it's so quiet until it progresses to AIDS. Now, the US actually went through an epidemic before things got better. So it started off in the 80s and peaked in 1995 when AIDS was the leading cause of death in adults between the ages of 25 and 44. When R was discovered and widely used, the death toll was lowered dramatically, as you can see from this graph that we've included, that the number of people living with HIV increased because people were living longer. People were still contracting the disease, but they weren't dying from it, and the death toll dropped off very quickly. As well as ART, the discovery of PrEP drugs, and lowered the transmission rates, and quicker, more accurate tests helped doctors keep the disease under control. But we hope that this has been an awesome video for you to learn a lot about HIV. It's a crazy disease and one that definitely deserves to be discussed. So we hope that you have a wonderful, wonderful semester, and thank you.